Hello, my name is Matt Green, and this is part 2 of the Fragile X syndrome lecture series. In the first part, we talked about what Fragile X syndrome was, what some of the phenotypes were. We talked about the genetic cause, as well as some of the animal models that have been produced to try and replicate the human condition. In this lecture, we're going to be focusing on how we've used those animal models and what we've found, some of the molecular changes in the animal models, and how we've looked at those molecular changes and tried to identify pharmacological targets to try and treat the human condition. So, as I mentioned in the last lecture, Fragile X syndrome is caused by mutations in the FMR1 gene. So this gene normally produces this protein, Fragile X mental retardation protein, or FMRP. And very generally, this FMRP protein is a negative regulator of translation. So, we have our central dogma of biology. You get from DNA to RNA. RNA is translated into protein. So FMRP normally inhibits certain mRNAs from being translated into protein, but it seems to inhibit a lot of mRNAs. So it's estimated to regulate about 4 to 8% of the total mRNAs in neurons. So without FMRP, these mRNAs aren't regulated, so they produce more protein than you normally would produce. And many of these mRNAs are important for synapse development and plasticity, as we'll see. So because FMRP does so much and it regulates so many different proteins, a lot of people have focused on different systems and seeing how these different systems are altered. Some of the observed effects of losing FMRP are upregulation of mGluR signaling, so the glutamate system is upregulated, as well as downregulation of the opposing inhibitory GABAergic signaling. So this whole balance between excitatory and inhibitory signaling is altered and shifted towards the excitatory signaling. So this is one explanation of why these the patients have increased sensitivity to seizure, for example. There's also downregulation of dopaminergic signaling, as well as increased oxidative stress to neurons. Just so we can, this is sort of the prominent leading hypothesis of Fragile X syndrome is this upregulation of mGluR signaling. So we'll go through this. So metabotropic glutamate receptors are G-protein coupled receptors. So very broadly, there's two types of receptors. So there's these ionotropic receptors on the left. So they bind neurotransmitter, and then they are permeable to some ion. However, these G-protein coupled receptors, they bind neurotransmitter just the same. However, they aren't permeable to ions. Rather, they're coupled to these intracellular signaling cascades that can then regulate ion channels. And they can do this by increasing or decreasing the activity of the ion channel, or they can actually remove or insert the receptors from the membrane. So I just wanted to talk very briefly about LTP and LTD, as this is important to the hypothesis of mGluR signaling in Fragile X syndrome. We could spend a whole series of lectures on LTP and LTD, but very generally and briefly, LTP stands for long-term potentiation, and this is the strengthening of synapses, whereas LTD is long-term depression, and it's the weakening of synapses. So one main way that you can strengthen synapses is by increasing the number of postsynaptic receptors. So if you start off in some basal state here, say you have 3 receptors, some stimulus comes in that induces LTP, and then it increases the number of receptors from 3 to 5. Whereas if you give the neuron some different stimulus that can induce LTD, you can actually decrease the number of postsynaptic receptors. And the reason this is important is because these mGluRs can actually induce LTD. So DHPG, this drug, I think 2 of the groups had this in the last, the endocannabinoid synthesis activity. It's an agonist of mGluR15. So when you activate these metabotropic glutamate receptors, this triggers some protein synthesis. Some of these proteins, you guys don't have to memorize, but they lead to receptor internalization, and thus LTD. So you guys might be able to predict where FMRP comes into play in this protein synthesis. So normally, FMRP prevents this internalization of AMPA receptors by being a negative regulator of protein synthesis. So in the normal state, if you have FMRP on the left here, say glutamate binds to mGluR5, this triggers this protein synthesis, and FMRP sort of slows the internalization of these AMPA receptors. However, in Fragile X syndrome, if you don't have FMRP, it doesn't halt the brakes on this process, and a lot more receptors are internalized, and the synapse is thus weaker. And this is sort of one of the main hypotheses for how you end up with the phenotype that we talked about in the last lecture, where there's these abundant immature spines. There's a lot of these weak synapses. It's difficult for the synapses to become mature with this overactivated pathway, and thus you end up with a lot of these synapses trying to compensate. So one thing that people have thought to do is, well, if you inhibit mGluR5 receptors, this will prevent this protein synthesis from occurring to begin with, and it'll try and compensate for the lack of FMRP. And thus you might be able to treat some of the symptoms. So there's a number of inhibitors, MPEP, CTEP, Phenobam, Mavoglurant, and we'll talk about more about these last two, Phenobam and Mavoglurant, in the next lecture, as they've been used clinically to try and treat Fragile X, whereas MPEP and CTEP have been used more on the preclinical side, trying to see if they can reverse some of the symptoms in mice. And indeed, if you look at the FMR1 knockout mouse model that we talked about in the last lecture, inhibitors of mGluR5 can restore some of the dendritic morphology. So again, these knockout animals have an increased number of spines. So the blue, the knockout that was given vehicle, have an increase in the spine density, so increased number of spines. And then if you treat these animals with CTEP, this lowers the number of spines and restores it to the wild type level. Additionally, inhibitors of mGluR5 restore some of the behavioral deficits in knockout mice. There's been a lot of behavioral paradigms that have been tested. It prevents seizures from happening. It restores some learning processes. This one paradigm I wanted to talk about because it's been one that's been used a lot in humans and animals, which is interesting. So not a lot of paradigms that have been tested like this. So this is called pre-pulse inhibition. It's very simple. If you give a pulse, usually it's an audio pulse, so a loud sound, it produces a startle response. However, if you give a pre-pulse, a softer sound right before the loud sound, this sort of primes the animals and lowers the startle response. And in a lot of disorders, Fragile X syndrome especially, other things like schizophrenia, it's been found that these patients the startle response isn't as inhibited as much if you give the pre-pulse. So you can see the PPI, the pre-pulse has less of an effect in the knockout. However, if you treat it with MPEP, this restores back to the wild type levels. So we'll talk about more in the next lecture about how these inhibitors have been used clinically with some of these same paradigms, but I wanted to introduce this PPI in this lecture. So we'll continue this in the next lecture, talking about the human clinical trials. Thanks.